Konnichiwa. Good afternoon. So I'm Richard Thompson. Our lecture today is entitled Discovering Microplastics, Their Environmental Accumulation, Impacts and Solutions. There are 3 of us laureates for this prize, and we are going to divide the lecture into 3 separate parts. The first section from myself is entitled Discovery, Environmental Distribution, Sources and Pathways for Microplastics. I want to take you really this afternoon on a journey of some 30 years of my life that was really unexpected. Back in the 1990s, I was training to be a marine biologist. I didn't imagine that career was going to be focused around people's rubbish. What happened was there was a chance discovery that took me from marine biologist to someone who has been described as the godfather of microplastics. I'll come back to that name later on. So how did it happen? The film touched on it. I was conducting experiments, marine ecological experiments, but every day rubbish was arriving. I kept cleaning it away, kept returning. So working with students, I organized beach cleaning activities, voluntary cleanup to remove the rubbish. I've seen similar cleanups just today here in Kyoto along the river, volunteers cleaning. Those volunteers were documenting what they found. They were putting it into spreadsheets, and we were looking at that data. As a trainee scientist, I was curious about the data. Something really important struck me was that the very small pieces, the fragments, were not being counted, they weren't being recorded, they weren't being removed. And yet, as you see from the photo on the right-hand side, they were actually probably the most abundant pieces in the environment. And so I asked a very simple question. I wondered, what are the smallest pieces of plastic in the ocean? And that was a question I put to some of my students, to look at samples from the environment and to see what they could find. It was that curiosity, that question, what are the smallest pieces of plastic, that set me on this 30-year journey. In 2004, 10 years later, we published the first paper on microplastics. Why so long? Because the work had been done largely in my spare time. Most of my career I was paid to still work on marine ecology. So this was kind of a hobby, working with student projects, trying to piece together the picture. What did we discover in that first paper? Well, we showed that microplastics were present in the seawater and in sediment all around the UK. We showed that their abundance, the quantity, had increased significantly over time. We showed that they could be eaten by a range of different creatures. That doesn't prove harm, but it proves the potential for harm, that a wide range of creatures were eating them. And this paper pointed to the potential for global accumulation and also the possibility of physical and chemical toxicity. And critically in that paper, we used for the first time the word microplastic in this context. So this graph is a plot of the publications that you'll find in the scientific databases of publications. In 2004, one paper. The next year, another one, and so on. But then suddenly you see this rapid increase. Today, the field of research has been contributed to over 4,500 publications globally on this topic. The team that are here today have contributed some of those, but this is an immense field, and the point that we're at today has come from global contributions, including amazing work done here in Japan. Some of our early papers then, to go back, one of the things we wanted to show was, is this phenomenon just in the UK or is it global? So here was the first global study where we showed that microplastics were present on shorelines all around the world. Everywhere we looked, we found it, mostly close to big cities. There were 1 or 2 locations, such as here, down in the Falkland Islands, that were fairly remote, but most of the work was in big cities. Each piece had to be individually identified. They're the spectra that you see on the right-hand side. They're too small to know that they're plastic just by looking at them in the same way that you might with a plastic bottle. I was curious then, okay, if we find it near to big cities, what about the more remote places? Then came inquiries from other scientists. A scientist called Rachel Obard emailed me and said, I think I might have microplastic in Arctic ice. Could you help? And we looked at it. the quantities shocked me. The quantities were greater than you might find in the water I'd been looking at close to the big cities. We later discovered that the physical properties of ice formation actually led to concentration of particulates. And so we published a paper showing substantial accumulation of microplastic in Arctic sea ice. We then went down to the deepest parts of the ocean. Well, not the very deepest, but we went 3,500 meters down. And we showed that microplastic was accumulating in substantial quantities there. Again, a long way from the human sources. More recently, we went nearly to the top of Everest, and we found microplastic there too. This truly is a global phenomena of contamination. But it's not just in the environment. It's also now, we've shown, in the creatures that inhabit our blue planet. Working with a student at the time, Amy Lusher, who now runs our own team of microplastic researchers in Norway, Amy showed that of 500 fish that we looked at, over a third of them had microplastic in their guts, in their intestine. She looked at 10 different species. Some of them were commercially important, species that we would eat. The plastic was clearly present, not just in the environment, but also in the species living in the environment. Again, it doesn't yet show harm, but it clearly shows the potential pathway that the microplastics from human society were getting into the environment and into creatures. What happened when these plastic particles got into the creatures? We showed early on in 2008 that for very small particles, they could pass from the gut to the circulatory system and then pass around the rest of the body. This is in a marine mollusk. And then more recently in 2018, we showed that for the nanoparticles, this transfer from eating them to the rest of the body would be incredibly rapid. The picture in the center there is after 6 hours in a fish tank containing what we believe to be environmentally realistic concentrations of nanoparticles. But then if you move the scallop to clean conditions, most of the particles, but not all, pass out. This is 8 days in clean conditions. And what does it tell you? It doesn't tell you yet about harm, but it tells you perhaps some of the tissues you should look at to look at harm, where the plastic is accumulating and staying in the hepatopancreas, similar to the liver in humans. It's accumulating and it's not passing out. This points to possibilities that will be picked up later when we come to talk about harm in the later part of the talk. So I want to move from the accumulation in the environment and in organisms to some of the sources now. Now, the very first thing that stimulated me to work on this topic, it clearly shows very substantial, a major source, if you like, is the fragmentation of larger items. The large items, the crisp packets, the bottles, the fishing rope and netting, breaking down progressively into smaller and smaller pieces. On top of that, we also showed that there were some direct sources of microplastic, where particles were entering the environment already very small. We showed, well, I say we, this was key work from a PhD student, Imogen Napper. During her PhD, she literally counted the small pieces of plastic in these cosmetic products. A single container could have 3 million plastic particles in it, particles that would inevitably escape down the sink, down the plug hole when you had a shower, or down the bath. This paper led to legislation in the UK, but also worldwide. And you think that's an amazing triumph for a young PhD student to produce this evidence that goes directly to Parliament and informs legislation. And it is a real triumph. At the same time, for me, it's a frustration. It's a frustration, why? Because when you look at the patent on the use of these small bits of plastic, that was filed by the industry 50 years earlier. And I have to ask the question, did nobody in the industry ever wonder where these hundreds of thousands of tons of plastic that they were putting in these products were going? If they had asked that question 40 or 50 years earlier, we might have been, if they collaborated with the scientists 40 or 50 years earlier, rather than waiting for us to find it, we might have seen off this problem decades ago. And it points to the future of extended producer responsibility, where science and industry must work together to safeguard our blue planet. We also showed that there were substantial quantities coming from textiles. Every time you wash a washing machine full of washing, hundreds of thousands of fibers could be released that are then also going to escape to the environment. But it wasn't just while you're washing the clothes. We subsequently showed that while you were walking around in the clothes, over a year, the emissions to the environment were similar, so that the pathways to the environment of the textile fibers, microscopic pieces, were from washing, but also walking around. And yet the first study we'd produced showed that there were very substantial differences between different garments up to an 80% difference could be achieved, reducing the amount of microfiber emissions by better design of the clothing. Just from some of the clothes that we currently find in the shops, they weren't designed for low emissions, but it clearly points to the role of industry in helping to reduce these direct sources to the environment by better design. A further source we examined much more recently was that of tyre wear particles. As you drive along the road, the tyre wears out, those particles regrettably go into the environment. We showed that the quantities were very substantial. They were passing from the road into the water systems when it rains. They were also passing via the water system to wastewater treatment. But in addition to that, a very substantial pathway was via the air. The particles were going up as dust into the air, they were blowing in the wind, and they were settling back down onto the Earth's surface, including the water surface. We're currently working together, Penny and I, to look at what happens next. How far do they travel in the environment? How harmful might they be? In addition to all our work on the coast, we also showed that rivers were a really important pathway to the marine environment. One of my colleagues once said, the sea is downhill from everywhere. So rivers actually are connecting places, potentially hundreds, thousands of kilometers inland, this conveyor belt, carrying rubbish out to the oceans, including the microplastics. Before I conclude, I want to really acknowledge some key work here in Japan by my good friend Shigi Takada, who's here in the audience today. Shigi had shown in 2001 that plastics could accumulate chemicals very rapidly in seawater, chemicals that were hydrophobic in nature. And so we took his studies and we applied that to the microplastics, and we showed indeed that the microplastics would rapidly accumulate chemicals from seawater, and that critically they would release those chemicals into the guts of creatures. It doesn't yet show harm, but it shows a clear pathway, not just for the particle toxicity, but also the potential mechanism for transport of chemicals. We're going to come back to that in a later part of the talk. So it was 2016 when actually it was a Member of Parliament, when Tamara and Penny and I were giving evidence on microplastics to the UK Parliament, described me as the godfather of microplastics, which was nothing I would have imagined when I was training as a student. If I look to the future then, just to conclude, I mean, this is the graph of the generation of plastic waste since 1950, where the graph becomes a dotted line here. All of this is the future. That's the bit that we've got the opportunity, if we choose, to influence. And I wish, and I continue to wish, and I think, how could I have changed things so far and prevented the rubbish that's entered the environment during my lifetime? This is a picture of me at my second Christmas with my mother. Even then, you can see that most of the toys around me are plastic. It's a testament to the durability of plastic that this blue and white plastic sailor over here is still with me today. His joints are better than some of my own, okay? That's the way we need to use plastics in durable applications. And yet 40% of everything we make in plastic is single use. It brings benefit for a few seconds, a few moments, but it persists as waste for centuries. What about when I first started counting the small bits in the environment? Well, the quantity of plastic had increased a bit by the early 90s, but it's nothing to where we are today. If I could have been a magician and stopped it then, everything to the right would have been prevented. When we published the first paper about a decade later, the amount had doubled again. If I could have stopped it then, again, everything to the right would have stopped. Here we are today in 2023. Quantities have doubled again. But still, it's not too late for our blue planet. All of this is the prediction for the future. This is the bit we've got the opportunity to influence if we change our ways and start to use plastics more responsibly. We're in the era of the UN Plastic Treaty. That is a fantastic opportunity, a once-in-a-planet opportunity, but we have to get it right. The science that's brought us here to this place today, defining the problem, is going to be just as important going forward as we start to define the solutions. Thank you very much.
Much, Richard. The next part of the talk is actually from Tamara Galloway. As you've already heard, I'm very sorry to say that she wasn't able to come today, but I am giving the talk on her behalf. So Tamara is a professor of ecotoxicology at the University of Exeter. Her research group at Exeter addresses three main research questions. How does pollution damage living things? What makes some species more vulnerable than others? How can we use this knowledge to protect the environment and human health? Just because microplastics are present everywhere in the environment, it doesn't mean that they are causing harm. For most contaminants to do so requires them to come into contact with the living things around them. In the case of microplastics, the reason they are such a cause for concern is they overlap with the prey item size of many different marine animals that rely on particulate matter and planktonic species for food. To address the question of whether this was happening required us to study a wide range of species depending on the nature of the question being addressed, right across the food web, from the ocean surface to the ocean floor. Initial studies were conducted using fluorescently labelled plastics in the laboratory because this enabled us to use microscopy to locate the plastics after they'd been eaten. In these images, we studied the uptake of microplastics by planktonic species. On the left, there's a copepod, Centrotopicus typicus, and on the right, a young 3-day-old larval oyster. In both of these cases, you can see that the animals readily ingested the test plastics and could be located right down to the tiniest of particles in the animal body. The next important concept to study was what happened to the microplastics. After they'd been eaten, did they pass straight through the body? In which case there would be limited opportunity for them to cause harm. The next stage was to do some experiments with mussels, filter-feeding animals, which were important both as a food source and for maintaining the clarity of coastal waters. We found that microplastics of different sizes could actually pass across the lining of the gut. From here, they could be taken up by the blood cells and therefore circulate around the whole body of the animal. In fact, the microplastics could still be measured in the animal bodies for over 48 days. At this time, the experiment was finished, but they could have been there for much longer. This important finding confirmed that microplastics were available to cause damage to the animals ingesting them and could also be transferred to any other animals that ate them. Tamara and her team also pioneered the use of another technique more commonly reserved for human health studies, known as hyperspectral imaging. This is where infrared lasers are used to vibrate molecular bonds, allowing them to be visualized. Here, the technique was used to locate the microplastics embedded within the lining of the gut of a crab. And this meant that we didn't need to label the plastics or dissect the animals to visualize them. The same technique has been used to locate microplastics stuck to the outside membrane of the gills of a crab, attracted here by hydrophobic nature. We also wanted to study where the microplastics may be ingested higher up the food chain. A shared PhD student, Emily Duncan, found microplastics in the gut content of 102 stranded turtles, including individuals from all 7 species across 3 ocean basins. The presence of microplastics with no ingestion of larger plastics suggests the possibility of multiple ingestion pathways, including polluted seawater, sediments, or trophic transfer from contaminated prey or foraged items. So, what types of harm might we expect to see from microplastics? Here, there was an experiment done with sedimentary marine worms. They were used as a model species to allow studying the effects of culturing the animals in sediments contaminated with particles of PVC for up to 1 month. The physical effects of the exposure were that the worms were less able to ingest their normal food and their energy due to the ingestion of the non-nutritious particles. The worms actually ate less, the food passed through their gut more slowly, and there were inflammatory changes visible in the worm's blood cells. But microplastics are complex contaminants, combining not just the solid plastic, but they also have many different chemical additives added to make them harder or softer, or as a UV protector or antimicrobials. In the next set of experiments, they measured the release of a plasticizer called phthalate from plastic particles into the worms. Phthalates are classified as endocrine-disrupting chemicals, able to adversely affect hormone and immune systems. Finally, they looked at a third concept of harm, and it's illustrated here through the transfer of absorbed chemicals onto the plastics. In this case, another form of microplastic fibres released from cigarette butts was used. These had toxic nicotine and cotinine on their surface and have led to the inhibition of burrowing behaviour in the worms. This function is crucial not only for the health of the worms, but also for the health of the marine sediments to keep them well oxygenated. It could be that contamination of the marine environment is again an issue only for the environmentalists and doesn't really affect the average person in the street. But there, I think you would be wrong. In this study of plastic polymers, present in a range of high-value seafood, they used analytical chemistry technique of pyrolysis gas chromatography to detect the plastic. And enough plastic was present to contaminate the average eater of a plate of sardines to around 30 milligrams of plastic at every meal. If you want to look at that in terms of numbers, some well-modelled estimates are that the average person consumes up to a credit card or 5 grams of plastic per week. But this isn't just from seafood, it's from packaging, from fibres, and even from the air we breathe, particularly indoors. So it's not ethical, obviously, to try to make one's students ingest an environmental contaminant to study the effects. But another approach is to remove it from their diet. So at Exeter, they enrolled 120 students into a trial of unpackaged and unprocessed foods to test how this affected their plastic intake. Regrettably, it proved impossible for them to find enough to eat that had not been packaged or processed in some way, illustrating the ubiquity of these contaminants that we have been studying now for many years. It also highlights the urgent need for solutions. So I pass now to my element of the talk, which is delivering evidence, raising awareness and evaluating solutions. So to start at the beginning, my background is in molecular marine biology with a particular focus on zooplankton. As such, ever since Richard coined the term microplastics, I've been really interested to determine if the prevalence of these small plastic pollutants in the marine environment cause a problem to the small but vitally important marine zooplankton. Zooplankton are common to marine ecosystems across the globe. And one group of zooplankton that has been my focus are copepods. They are amongst the most abundant animals on our planet and provide a key link between primary producers such as the plants and higher trophic levels such as fish. They also help to regulate the Earth's climate through their role in the global carbon cycle. Our initial studies on zooplankton were undertaken by a joint PhD student between myself and Tamara, Matthew Cole. And he looked at zooplankton. They were based on quite basic laboratory exposures, performed when we had little knowledge of environmentally relevant microplastic characteristics or their abundances. These studies have shown that every species of zooplankton that we have looked at have the capacity to ingest microplastic. 15 years down the line, and our experiments have become much more sophisticated and environmentally relevant. We now have a much better understanding of the bioavailability and impact of those microplastics, not just to zooplankton, but to a whole range of other marine animals. In summary, we know that zooplankton can ingest microplastics, And when they do so, they exhibit reduced feeding capacity, a decline in energy reserves, and lower reproductive output. This has direct relevance to both the animal populations and the communities. We also now understand that the chemical profile of those microplastics ingested impact on the health and may act as endocrine disruptors and impact molting. Microplastics ingested by zooplankton are also egested in their fecal pellets, and this has been shown to alter the properties and sinking rates of the pellets. Copepod fecal pellets are very important because they help remove carbon dioxide from the sea surface as part of the biological pump. Therefore, a lower sinking rate increases the opportunity for the pellet to be eaten by other organisms, but also reduces the organic matter that reaches the sea floor. We've also shown that it's not just in the laboratory. Microplastics have been shown to be consumed in the natural environment, and here there is evidence they may actually select what type of plastic they eat. However, as one of our PhD students showed, Zara Botterill, the factors affecting the bioavailability of microplastics to zooplankton are complex. And laboratory-based studies have confirmed that the shape of microplastics influence their uptake, but that this varies with different zooplankton. In addition, infochemicals such as DMS infused to the surface of microplastics may increase their uptake. Only with combined laboratory studies and in situ assessments can we better assess the current level of risk posed to the marine environment and tailor mitigation strategies to have the greatest impact. While our work on microplastics and zooplankton continues, it's expanded into many different areas, from the tiny zooplankton up to marine animals. We work with colleagues across all science areas, including chemists, looking at how infochemicals may make that plastic tasty, working with modelers, determining where the plastic goes and where the hotspots are. We've worked with remote sensors, detecting plastic litter from space, including microplastics, and social economists, calculating the social cost of plastics. As we have been leading the way in this field, we have had to be innovative in designing novel techniques. For example, from designing the sediment microplastic isolation unit as a tool to remove microplastics from a range of sediment types. This was developed by Rachel Kopok as part of her PhD, to the development of a non-invasive methodology pipeline to investigate dietary exposure of seals to microplastics, again done by Sarah Nelms during her PhD. Our expertise has also involved nature-based solutions, and it is on these solutions that the rest of the talk will focus. Over many years, we have built up the information, as Richard has said, of the source, the distribution, the prevalence, the bioavailability, and impact of microplastics. But more recently, we are using this knowledge to help develop solutions. One such solution to remove microplastics is a novel approach inspired by nature. While large plastic can be cleared from waters and beaches, microplastics are so small that it's really difficult to use mechanical mechanisms. So during our research, we have found that mussels are relatively tolerant to marine pollutants. They're voracious filter feeders, natural biofilters that can improve water quality, removing excess nitrates, phosphates, bacteria, and particulates. So for this project, instead of focusing on plastic toxicity, we wanted to scope whether there may be a role for mussels to act as a nature-based solution, helping to remove microplastics from polluted waters. We know from field sampling and also from lab experiments that mussels can filter out microplastics. But what we wanted to know was how efficiently can they do this in flowing water. So this was tested in a custom-built flume tank, where 5 kilograms of mussels, as you can see here, were added to the tank, filled with filtered natural seawater with a mixed algal diet. Then we added nylon fibres and polystyrene beads as representative microplastics. Following replicate exposures, we demonstrate the rapid removal of both polystyrene beads, as you can see on the left, and also of the nylon fibres, shown in the middle, by mussels over the first 3 hours of exposure. After this time, the removal rates did tail off, likely as the mussels were saturated with food. So the results from this showed that 5 kilograms of mussels can remove 250,000 microplastics from flowing water in just 1 hour. Encouraging news for our nature-based solution. So the next phase was to use information from the lab and flume work to develop a system to deploy the mussels and collect their feces in the natural environment. Here, there would be lower microplastic concentrations. We used a cage system whereby the mussels can't escape. They're also protected from predation, but there is a flow of water allowed to pass over them. So 5 kilograms of mussels resulted in 4.5 times more microplastics captured than the controls with no mussels. This showed us that there is certainly scope to expand our testing to multiple sites with a greater amount of mussels in a stacked system. So such a nature-based solution as this could help clean up and stem the flow of microplastics to the open sea. It should be prioritised as close to source as possible, where there is a threat to coastal ecosystems, organisms and human health. However, prevention should certainly be the first priority. Another solution at source is to work towards a circular economy. The linear economy of to take, make and waste means that a large volume of plastic is discarded after use. Not only is this causing an environmental problem, but it is also a huge loss of valuable resources. We need strategies to prevent leakage into the environment, reduce production and consumption, and to support reuse, repair and repurposing. Recycling should be seen as a last resort or a later resort to decrease the waste generation and pressure on waste management systems. We hope to inspire innovation to consider the whole of a plastics item's life, from the design stage all the way through to the end of life, with the intention of prolonging the lifetime of products. We need to drive towards a circular economy that maintains plastic at their highest value for the longest possible time. Another possible solution to the problems of plastic waste accumulation is offered by biodegradable bio-based plastics. These are made from renewable carbon, such as plant material, and as a consequence of their molecular structure and resulting properties, they are regarded to have enhanced rates of biodegradation compared to conventional plastics. The BPPs are already widely used in applications with substantial pathways to the natural environment, such as agricultural mulch film or textile fibres. Yet our understanding about their fate in the natural environment is actually poorly understood. We're currently improving our scientific understanding about the fate and impact of biodegradable bio-based plastics in the environment. We hope that the outcomes of this research will provide the potential risks and benefits of these new plastics to help guide development for the next generation of innovative plastics that are environmentally safe, non-toxic and not reliant on fossil fuels. Working towards solutions for plastic pollution, we have been raising awareness and giving evidence. We are passionate about disseminating our research to a wide audience, supporting the next generation of scientists and creating widespread awareness. Our pioneering work has reached a global audience, featuring in numerous programs, including Eating Our Way to Extinction, Plastic Warriors and Blue Planet 2. By raising awareness with the general public, we help to connect people's everyday choices with the impact plastic waste is having on our oceans. Collectively, myself, Professor Galloway, Richard Thompson, have presented evidence to a range of bodies, from the House of Lords to UN hearings on microplastics. This has contributed to the plastic bag levy and a ban of products containing microbeads in the UK, Canada, New Zealand, US, and other places. Legislative changes are very welcome, and a clear sign to the general public that initiates behavioural change. However, legislation to date has been minimal and far more needs to be addressed. More recently, we are contributing towards the International Negotiating Committee of the high-profile initiative of the Global Plastics Treaty. This is bringing the need for global cooperation and collaboration into the foreground and has the potential to influence positive change at all levels of society. We hope the treaty will be based on independent, transdisciplinary evidence and to include discussion across sectors, including scientists working with industries, NGOs, governments, local communities and world leaders to initiate appropriate solutions for a global scale change. We are the generation shaping the future of our Earth. Our choices and actions determine our impact on the environment. And as Richard said at the start of the talk, this is our blue planet. Its future and our future is in our hands. Thank you.
Richard, Penny, thank you for your presentation.